It says we're live again. It's on the top corner. It's yellow. It says it in black. Do I believe it? Possibly. It might be live, but I don't think I am. Anyway, this, and I've done a JD Kane again. I don't believe it. Anyway, right. This should have started at 11 o'clock tonight, and it should have been last night. But I lost track of the days of the week. And then tonight I fell asleep and didn't wake up in time. So I'm actually not that long awake and very, very woozy. But anyway, welcome everybody. Here we are. We decided we'd do a wee hangout about the flu because there is a bit of a flu epidemic at the moment. It's hit Australia, it's hit the UK, it's hit the US. So we thought, let's talk about flu. So what is flu? Well, we all know it's an infection that you get. The interesting thing is what the infection is by. And it's that all of the rest of it will then flow. Why does it do what it does? Why is it such a problem? It all comes from what it actually is. So we start off with the fact that it's a virus. But it's quite an unusual wee thing. It's one of those viruses that seems to have managed to encapsulate everything that a really horrible virus needs to encapsulate. It's an RNA virus for a start, so its genetic material isn't DNA, it's RNA.
Mm-hmm.
And
Fiona, I don't know if you wanted to share a picture of what you're talking about so people can kind of have a visual aid.
Well,
maybe pull up a picture of a influenza virus and an EM or just a diagram.
Yeah, that's what I'm trying to do at the moment.
Yeah. And as a virus, it can only reproduce itself once it is within the host cells, not just within the host, but within the host cells.
Yep. Okay, let's see, application window. There we go. Right. Has that popped up?
Yep, see it fine.
Right. So it's a single-stranded RNA virus. It can occasionally be double-stranded, but that is one of the unusual subtypes. I think that's influenza C.
Mm-hmm.
But it's a single-stranded RNA virus. And it's a positive strand. And I think Ian can get into that and explain it to you possibly better than I can in my very sleepy state. But if you look at the little diagram, you will see these little red coily things. Now, they're supposed to be the RNA genome, and you'll notice there's more than one of it. And that's because its genome is segmented. Now, put a wee pin in that, because that becomes very relevant later on.
Yep.
When we start talking about antigenic shift and antigenic drift, and we will explain what they are. But the fact that they've got, it's got a segmented genome like that, that's one of the reasons why it is such a problem. Now, the flu itself, this is the virus particle. And on the surface of it, there are two really important little proteins, hemagglutinin and the neuraminidase. So that's the H and the N. And you can raise antibodies to these. Not all hemagglutinins are the same, and not all neuraminidases are the same. And so the different types of hemagglutinins have been given numbers. 1, 2, 3, etc. And the different neuraminidases have been given numbers. And you can use antibodies specific to, say, H1 or N3 and see if they work against the particular strain you have. Once you identify those antibodies that will bind those, you've typed your flu. It's an H1N1 or it's an H2N3.
Can I mention a brief explanation of antibodies just for any laypersons in the audience? That an antibody is a large, sticky, but specifically sticky protein that your immune system makes that recognizes and binds to things that your body recognizes as foreign. By doing that, that foreign entity becomes marked for destruction by the immune system. And if antibodies are stuck all over the hemagglutinin and neuraminidase of flu, it can no longer enter the cells of the host and so cannot reproduce.
Yeah, because it's the hemagglutinin that allows it to bind to the host cell.
Mm-hmm.
And it's the neuraminidase that allows it to bud off at the end. Because the neuraminidase, doesn't it cleave a sialic acid residue?
That's right.
And the host cell, we'll get to explaining why the host cell membrane is very important. But anyway, so that's the very basics of, here is the flu. This is how we type it. And the segmented genome business is why we have all sorts of problems with it. And we'll explain that further down the line. I'm going to stop sharing that wee picture because I need to look up other stuff.
And as a brief comment to G-Man et al., there is no faith required in the data and commentary that we'll be offering, because all of this is a function of very detailed and long-standing scientific and medical research driven by data, not faith.
Yes. If at any point as we do this, if anybody in the live chat doesn't understand something or has a question, please type it into the live chat. Because with me being so sleepy, I'm hoping the guys are going to carry me in this hangout. Mm-hmm. Now, keep an eye on the live chat and any questions that you have, try and get to them, okay?
I'm also happy to provide citations and explanations of those citations in response to those questions, although not live as we're broadcasting. Mm-hmm. So I'm happy to back up anything that I say, and I suspect anything that is said, with the appropriate level of peer-reviewed publications.
Yeah. Right, so that's the basic idea of what the flu is. So as Ian said at the beginning, when we get flu, the virus cannot do anything until it gets inside a host cell. So it sticks itself on to the membrane of the host cell. I've got another diagram. I'm just using these straight from Wiki because they're actually really nice diagrams.
Well, Fiona, you're welcome. I contributed to that webpage.
Cool. Right, here we go.
And it may be of value to outline at least our levels of expertise on this. I'm aware that Fiona has a PhD in microbiology. Fiona?
Yeah, bacterial physiology.
Bacterial physiology. My doctorate is biochemistry, and I specialize in vaccine and biological drug development, and have done for the last 20 years. Right. And RSC, you're a medical professional yourself, I understand.
Yes, I'm a doctor of internal medicine. I practice right now in Cleveland.
Okay.
Cleveland, that's where you end up getting to if you're trying to navigate to New York without that guy in The Day After Tomorrow, isn't it?
That's right.
It's the best line of the whole movie. Without me, you'll end up in Cleveland. Can anybody see that picky?
No.
Nope.
I know why. I didn't say share. Picky.
Beautiful.
Right. What did it? Right. This is what happens on a very superficial level when a flu virus particle meets up with a host cell. So this big pink blob is your host cell.
Yeah.
And this little thing that has little feet all over it is your virus particle.
Let's just stress that this is not a scale diagram.
Yeah, this is not a scale diagram, definitely not. This is a cartoon sort of thing, right? So what happens is that courtesy of these molecules on the surface of the flu, they're able to attach to the outside of the cell. And the cell then endocytoses them. It goes round about on either side with little, what you would call pseudopodia, right?
Mm-hmm.
And these little pseudopodia go all the way around and meet at the other side. And so now your flu virus particle is in a little vacuole inside of the host cell. So it's got there. It's nearly there now. It's in the castle, but it hasn't reached the Queen's chamber yet for a bit of rumpy-pumpy. It now has to get its genetic material into the nucleolus of the cell, where all the bits and pieces happen with nucleic acids, whether it's replication, transcription, translation. That's where the cell would be doing all of that. So it releases its contents, and that's, see all the little RNA strands in it? They get poked in to the nucleus of the cell. And it's at that point that the machinery of the cell gets hijacked. So now this poor cell is, let's call it brainwashed, into making, instead of its own messenger RNAs that it needs for itself, it starts making nucleic acid of the flu, making lots and lots and lots of copies of it. And the other thing that the virus carries inside its little capsule here, see, is an enzyme called RNA-dependent RNA polymerase. It carries that enzyme with it. Because what that is able to do is use the RNA genome of the flu virus to make complementary strands of RNA. Now, you all know that normally you would go from DNA to RNA. On this one, you're going from RNA to RNA.
Fiona?
Uh-huh.
Good time to comment about why RNA polymerase is an interesting factor in terms of flu changing so quickly.
Yes. But just let me finish this one wee bit. So it's making copies of it's making complementary copies of itself. And this is a positive sense virus. So it's going to make all of the things, using these, it's making all of the things it requires to make new virus particles. So that will be the components of the shell of the virus particle, the hemagglutinin, the neuraminidase, etc. It's making them, and it's also making copies of its segmented genome. And it also, from its own genome, translates more RNA-dependent RNA polymerase. So it's made all these components, and then these all get packaged up into a little, if you see a little sort of packagey thing.
Mm-hmm.
Now, the hemagglutinin and the neuraminidase that it's been making, they're being inserted into the membrane of the host cell and sticking out. When this little package of the new genome copy and the required enzyme is ready, it goes to the membrane and it buds off. So it's now stealing the membrane of the cell. And the cell has all of these hemagglutinins and neuraminidases on its surface. And when it finally buds off, there it is. Not only has it been busy hijacking the replication machinery of the cell, it's stolen a wee bit of its membrane as well. So after a certain amount of time, the cell will die. Now, you also have to remember that because, as Ian said before, these things on the outside of the virus are antigenic. They're recognised by your immune system as foreign, but they're stuck on the outside of your own cells. So if your immune system is up to scratch, it's going to start battling, and it's going to be a battle between the virus itself trying to spit out enough virus particles fast enough, and your immune system coming along and killing your own cells. Because remember, it's now recognising them as foreign because of what's on the surface. And so that's why when you get the flu, you can end up with the lining of your lungs stripped off because the cells are dying anyway because of all the budding off of the virus particles. But your immune system is also attacking those cells because of these foreign antigens on the surface. And the optimum temperature for replication of the flu virus is 37. The optimum temperature for a cold virus is below 37. That's why it never spreads further than your upper respiratory tract, your nose, because that's slightly colder. But the flu virus can get deeper into your body because its optimum temperature is 37. So anyway, that's a sort of a little overview of what the flu is up to and how it does its dirty little deeds to you. Now, Ian was going to bring up why the segmented genome and why the fact that we're looking at an RNA virus that is using RNA-dependent RNA polymerase is very, very important when it comes to considering the flu and the strains of the flu. Take it away, Ian.
Okay, thank you, Fiona. That's a really nice introduction to virology generally and influenza in particular. The critical point here is how the RNA-dependent RNA polymerase works and what functions it does and does not have relative to DNA polymerase. RNA-dependent RNA polymerase is an effective enzyme, but it lacks what's called a proofreading and correcting function. What that means is flu copies itself very poorly. What does that mean? It means that the progeny flu viruses generated from an infection are genetically different at a faster rate than the parent strain. In a global sense, that means flu is genetically unstable. It changes very quickly under the influence of drift and selective pressure. And what that means is one vaccine at one time will not fix or prevent flu all the time. It's a moving target. Now, the segmented aspect of the genome is also interesting. The reason for that is it enables flu to recombine very quickly, both between strains in one species and critically between strains infecting different species, for instance, avian and porcine or swine flu. And that introduces a significant amount of novel genetic material to which the human may or may not form an effective immune response. And this is how pandemic strains and extremely pathogenic strains have arisen in the past.
Keep going, I'm rolling a cigarette.
She's rolling a cigarette. Maybe, I mean, we've covered a good bit of the virology and the reason that flu is a rapidly moving target. So to bring this to a more focused point for the audience, maybe RSC, you could discuss the clinical indications and the significant risks associated with this infection.
Sure, I'd be happy to. Like Ian and Fiona have said, this virus, influenza, is a very rapidly changing and contagious virus. I don't know if we touched on some of the reasons why it's so contagious. And in those pictures that Fiona was showing, most viruses are just composed of protein and DNA or RNA. But what really sets influenza out is the fact that it's covered by a layer of lipids. Basically, it's a layer of lipids that come from the host organism, which in this case is us, humans. The fact that the outer covering of the virus isn't just a protein capsule, but covered in this lipid layer is what gives it that extra virulence. It allows for the influenza to attack with much more potency, and its main target, like Fiona said, was the respiratory system. So
that's the reason it's got those lipids around it, is because it's budded off and it's nicked a piece of the host cell membrane.
Yep.
Because
it's basically stolen it.
The cell membrane is a lipid bilayer. That's why it's good, and it doesn't dry out as fast because of that. Sorry, just thought I'd throw that in.
No, thank you. It was an excellent mention there. I was just going to move on to the symptoms that people get from influenza. It's very typical: fever, sore throat, things that are related to your respiratory tract. So if you get like runny nose, if you start having a cough, if you have respiratory symptoms like you're having to breathe real fast, watery eyes, things like that are the hallmark signs for doctors clinically. We look at that, and that really tells us to consider influenza. Of course, the fever and the muscle aches and pains and other things aren't caused directly by the virus, but more so that the body is fighting the infection. Unfortunately, there is some fallout, some friendly fire when that happens, when your immune system takes over. In terms of the reason why this strain has been so impactful, I think I was talking with Ian before we started here. We didn't do a very good job here in the US preparing people for this strain. We were aware that Australia, South America, other areas had very bad flu seasons, and I don't think we necessarily prepared, whether that's governmental reasons, budgetary reasons, or whatever. We'll have to say that for another time. But that's probably the first reason. The second reason this strain is particularly impactful is it's not just causing flu, it's causing flu and allowing for other things to attack the body at the same time, particularly pneumonia, bronchitis, other infections of the respiratory tract not by viruses, but by bacteria, have been the main culprits of why people are expiring from this condition. Regardless of the age, they always say flu is very particular for the very young and the very old. But this year, we're seeing a lot of people of average age who are dying in 1 or 2 days. The average incubation period for this virus is only 1 or 2 days. So it acts very quickly, and you don't really have a lot of time to get the treatment you need once you begin to experience these symptoms. I can tell you that patients who come in with these symptoms, It's not uncommon for them now at this time to come in one day with the symptoms and the next day they could have septic shock. Septic shock is when
I was just going to say that's one thing. I've been reading about deaths from septic shock, and that's one thing I really wanted you to explain how this is happening. So I'm glad you got there. That's all I want to say.
No, thank you. Well, the septic shock is basically when you have an infection that spreads into the blood. Once you have bacteria in the blood from an infection like a bacterial pneumonia that has used the opportunity of the influenza infection to creep in and take over, it spreads into the blood. And once it spreads into the blood, it causes a vast array of problems. The immune system sometimes gets out of control. And what really happens is blood pressure falls. People who have septic shock, they are unable to keep their blood pressure high enough to keep their brain working. And when this happens, this is where death comes in. Septic shock is a very, very, carries a very high mortality, even if you're in the hospital, even if you're getting the antibiotics, even if they're giving you all the medicines and then they're pumping you with IV fluids and everything. It's still a very dangerous condition to be presented with. And because it spares no one, regardless of your age, you're seeing people of all ages affected by this.
Yeah, I just read about a man in Texas who's had both legs amputated and 9 of his 10 fingers because of septic shock from the flu. That's
correct. I've actually not been following the flu very much in Cleveland, but I have been following it back in Dallas, where I'm from. It's very sad for me to report, but there have been over 100 deaths just in the Dallas area this year. So we're just talking about 2018. That is an incredible number, considering that last year there were about 30 deaths in total. And the flu season isn't over.
I think there is a common misconception among the general public that flu is a disease that you get, you're sick, then you get better, everything's fine. It's an inconvenience more than a public health challenge. And I know that Jade had looked into some current numbers on mortality and also has information about the pandemic manifestation of flu.
Right. Well, one thing, there's a link here that I put on the inside, if maybe Fiona or somebody could put it on the outside, to a New York Times article. And I don't screen share worth a darn, so perhaps someone could screen share this, the graph here. But what I wanted to get to this, because this is, in the New York Times article, they're talking about how bad this flu season is, and it's more intense than any since the 2009 swine flu pandemic. It's still getting worse. And one of the things they said is it's now on track to equal or surpass that of the 2014-15 flu season. In that year, the Centers for Disease Control and Prevention estimates 34 million Americans got the flu, 710,000 were hospitalized, and about 56,000 people died. So a lot of people have this idea, oh, you just get the flu, you get over it. In 2014 to 2015, 56,000 people died from the flu. And they're saying they're expecting those sorts of numbers now. However, if you look at this little graph, I don't know if you guys can see it. Let me go down to it. My system's slow. It shows how this flu season compares with recent ones. All of the other lines here, the gray lines, are 2013 to 2017 flu seasons. Look at the red line. That's us so far. We far surpassed previous flu seasons. So this is scary. And one of the things, there's that. So this is scary. And so one of the things I also wanted to ask is, actually, I think Fiona can probably talk more about the pandemic in it started 2017. The great flu pandemic that started after World War I, which was absolutely devastating. And the number of people, the percentage of people it killed is just huge. But it also, interestingly enough, targeted people who were normally very healthy. Fiona, you there?
Yep, that's correct. And there were between 50 and 100 million deaths as a result of that particular pandemic.
Yeah, the pandemic was.
I would.
Sorry, Jade.
I was just going to say, so when people think that the flu is just harmless, you get over it. Remember those 56,000 deaths, and that's just here in the United States in 2013. 13-14 flu season. And remember that at one time there were just millions and millions of deaths all over the world due to a pandemic. And yes, we could have another pandemic. So Fiona, take it.
Yeah, I was going to say that if we go back to the pandemic that everybody knows about, the 1918 one, that was called Spanish flu. And it was H1N1 subtype. What that one did that was so lethal was, firstly, we had loads of troops returning from the war front and scattering all over the globe. And they think they took it with them. This is why it spread so fast. But the other thing it did was, it was one of those versions that the problem you had with it was not directly down to the virus, but down to the reaction of your immune system to the virus. You got an overwhelming immune response. Now, from the symptoms that were described at the time, they think that what the patients suffered was what they call a cytokine storm. They can be lethal. And that is when the immune system just goes, wah, and produces such a strong response. So the temperature absolutely rockets. You've got complement cascades going on through the body, all the rest of it. And these are the things that start to drop your blood pressure, that start to help with capillaries beginning to leak. And one of the symptoms that was common in that the 1918 pandemic was bleeding from just about every orifice. There was bleeding from the eyes and the nose and the mouth. And the lungs would bleed. They would also fill up with fluid. And these patients drowned. And this was because it was their own immune response that was doing that to them. So those with a healthy immune system were actually the ones that were most at risk. Unlike with normal flu, where it's those with a depressed immune system, like the very young and the very old, that tend to suffer the worst because of the damage done directly by the flu, because the immune system doesn't quite get on top of it as fast. So the amount of damage obviously done is greater. But in this 1918 pandemic, it was the patient's own immune system that caused the ultimate complications and death. And in fact, just as an aside, people will remember there was a drug trial carried out in the UK. I can't even remember what it was for. And 8 individual men were given an experimental drug, and they all got so ill they ended up in intensive care. And that was a cytokine storm. Their body reacted to what they'd been given. It turned out that a 1,000th of the dose would have been okay. Mm-hmm. And that was found out in subsequent clinical trials. But that's what a cytokine storm can do. The drug that they were being given wasn't going to do anything terrible to them. But because they were healthy individuals, their immune system caused them to end up in intensive care because it was just an overwhelming response. So that's what happens with some of the versions of flu. The 1918 pandemic is one of the classic ones. The other respiratory virus that did that, where patients would actually look as if they were getting better and then suddenly crash, and it was overwhelming immune response, was SARS If anybody remembers the SARS outbreak.
Yep.
Those patients who died because of the overwhelming immune response to the virus. So that's, well, put it this way, the flu doesn't intend to do that to you. All it wants to do is make more copies of itself. End of story. But if your immune system decides to take a hammer to crack a walnut, then it might crack more than the walnut and crack you as well.
That's right. And to stress, none of this, or a very small proportion of this, can happen without the establishment of an effective and productive infection. So avoiding the flu virus getting into your cells is the key way of preventing any of the subsequent symptoms.
Yeah.
Yeah, and I think that's, I was just going to say, it's something we need to talk about, but I wanted to mention a couple things is about what Fiona said. Yes, World War I, we had people all over the world coming home, going different places, spreading it. And let's not forget that today, the entire world is connected via air travel.
Yes.
And it'd be very easy for things. This is why, you know, when the Ebola virus came out, people were cracking down on you know, passports, for not allowing people to travel from certain countries and so on. We've seen these things happen. I can tell you about how cholera almost entered the United States some decades ago, but through LA, but they managed to stop it in its tracks. But what if they hadn't? Very, very quick response by not only the CDC, but the LA, you know, health authorities. The other thing is I've read a lot of accounts of what the pandemic did to communities in 1918. And just to give you, now you remember it's mostly healthy adults that were killed. I read a story that a man wrote. He was about 10 years old when the pandemic hit. And he wrote about what his life was like before the pandemic hit and how he'd play with all his friends and they'd play all these games and everything. By the end of the pandemic, he was the only one alive.
Mm-hmm.
All of his friends died in the pandemic.
Yep.
And that just, there were entire communities that were completely devastated by this pandemic. And so the human cost, I know we look at the numbers, you know, you say, wow, this is horrible. All these people died, just as like the thousands, tens of thousands of people who died in 2013, 2014. We need to remember these are real human beings. And this has a incredible effect on entire communities and entire families. Think about this man. If he survives his septic shock, he now has no legs and one finger.
Yep.
That's devastating. But yeah, so we really need to know, okay, we need to know about the flu shot, and we also need to know about how to keep from getting the flu, how to protect ourselves besides getting a flu shot.
Okay, I'll make one comment here as well. We have historical pandemics that Jade has described elegantly. The two things that are required for a flu vaccine to become a pandemic strain are sufficient infectivity and sufficient pathogenicity. Too much or too little of either one of these tends to result in an infection that can be severe but limited. Flu, as I've mentioned earlier, is a rapidly evolving, moving target. The likelihood of an additional pandemic occurring is almost 100%.
Yeah, I'll just explain one wee bit of that. You were talking about, you know, it's this balance between infectivity and pathogenicity.
Yep.
If it's not very infectious, i.e. it's hard to pass from person to person, then...
Sorry, you're muted, Fiona.
Is that any better?
Yep, you're fine now.
Right. Yeah, that's better. That's better.
If it's not very infective, so it isn't able for every one infected person, okay, if it only infects one other person, then you would just get a little flat line, really. It has to be above that. And I think, is it this year's season? It's 1.4.
Yeah, that's right. So every case generates more than one subsequent case,
Yeah.
which means you have an increasing number of cases causing more cases, and that builds up.
It's a snowball effect.
Yeah. And also, if it is very pathogenic, I mean, if it hits you and flattens you straight away, then you don't get a chance to pass it on.
Yeah. There's a historical study of a particular outbreak which resulted from swine and ducks being very close together.
And there, missus.
Yeah, I mean, the swine were raised in cages above the duck pond, fertilizing the water and so on. Great idea. There was a trans-species recombination event, and perhaps thankfully, everyone in that rural community died. They were all infected, they all died, therefore the strain didn't go any further. And that's one of the reasons that the horrific illnesses like Ebola tend to produce outbreaks rather than pandemics.
Yes, and like you said, the effect of pandemics and epidemics and what we're looking at preventing is the spread of this contagious disease to levels that basically go beyond what we're capable of controlling, what we're capable of handling, medically speaking. There is a limit. We don't have enough resources at this time to cover if, I don't know what you said, like if it's 15% of the population becomes sick, we don't have enough resources in the healthcare system to manage the outcome of that. And that's what's really scary. There is a tipping point that we have to be wary of.
That's right. And I mean, certainly within the context of the UK government, contingency plans are in place for exactly this kind of thing, such that loss of 30% of the productive members of the population tends to result in an infrastructure collapse in terms of healthcare, education, law enforcement, undertaking, food production, and so on. And this can happen relatively easily in the face of a debilitating, potentially lethal, and highly infectious disease. And although it's not the same disease, the impact of bubonic plague in Europe during the medieval times killed between 30 and 50% of the population, and it revolutionized the entire culture. There was a necessity to rebuild, and the socio-political effects of that are still being felt.
Yeah, can I just mention something about that? I've read about that too, because I find the subject of epidemics interesting and terrifying. And the bubonic plague especially interests me because, you know, it's endemic here in the West. Rodents have it. We get cases all the time. It's not a big deal anymore because we have antibiotics. And it rarely goes pneumonic. There's usually the bubonic and occasionally the septicemic form from people who are, like hunting and killing rabbits and things like that. And they get the septicemic form by cleaning the animals. But after the plague hit, there was there are people who chronicled what happened during the plague. We all know about Samuel Pepys in London, but there are people who chronicled it elsewhere. People traveling from place to place. And one of the things they pointed out was that one there was a Italian villa where some very, very wealthy people lived, lords and ladies, and they were used to having servants. When the plague hit, the servants left, and the people who were visiting the area came by and found that here was the lord and lady of the manor out there with sticks, poking the sticks in the ground, planting seeds to try to support themselves.
Yeah.
Because it affected everything. The servant class disappeared to a certain extent, and those who lived could bargain for their their work.
That's right. It had a massive impact on the cultures that experienced it. And this was, to get back to flu, a highly infectious and highly pathogenic organism. In the case of bubonic plague, a bacterial pathogen. But the analogy is exactly correct with regard to an illness like flu.
And it was the pneumonic form, which was spread by coughing. It was spread, well, coughing or just, because it got in the lungs. That was the one that was the most infectious form. And that was the one that spread fastest. The septicemic form comes from handling dead bodies, which includes handling, I said rabbits and things like that.
Yeah.
The bubonic form is the one that I think there's some controversy now over whether it was spread by fleas or by ticks, not ticks, lice.
That's right. And again, to comment and perhaps poke fun at the anti-evolutionists, the organism that causes bubonic plague doesn't cause horrible disease in the animals that naturally carry it, in the same way as the Ebola virus does not cause horrible disease in the organisms that naturally carry it. When it is transferred to a different species who is not evolved to manage it, it tends to kill us.
Yeah, which is why it's endemic in rodents. What that means is endemic means basically that it's always present in a certain percentage. So rodents and lagomorphs, because rabbits, you could argue whether they're rodents or not, but rabbits and hares also carry the plague virus, Yersinia pestis, which I always mispronounce, and so do squirrels and ground squirrels and mice and rats and things like that. But yeah, so if someone goes out hunting and they see a perfectly healthy cottontail rabbit and they shoot it, thinking, I've got rabbit for dinner, and then they clean the rabbit and whoops, they get the plague because the rabbit had plague. But you wouldn't know it. Perfectly healthy appearing rabbit.
Equally, again, this is a commentary to G-Man, who doesn't believe that HIV is a naturally occurring thing. The genetic similarity between the simian immunodeficiency virus and the human immunodeficiency virus is very clear and supports the cut-hunter transmission theory, such that a disease endemic within a population of non-humans that is a virus and copies itself poorly can easily transfer into another species where its symptomatic array may be significantly different, as in this case.
Well, and the Ebola virus most likely came from hunting and eating bats. I think, wasn't it bats?
I think that's the preferred hypothesis at the moment. Yes. The precise animal reservoirs are not fully understood.
Right. And there's another disease that I can't think of the name of right now that they also think of. It's what's called bushmeat. When they say bushmeat, basically you're just eating, animals from the wild. And I think bats are figured in another disease that I can't remember the name of right now. But in these areas, there are people who eat a lot of wild, animals. And that's what happens. It does transfer.
Yeah. So we've talked about similar diseases to illustrate points that are relevant to flu, just to be clear.
Right. Yeah. So how about, how does immunization work?
If you guys
are we there yet? Okay, go ahead.
If you guys have wrapped up on that part, I did want to mention Lusty asked a really good question in the side chat, asking about why is the flu season in this one small time window of the year? Why don't we see this disease happening, in the summer months, in the spring months, and at other times of the year?
Yeah.
I gave him a quick question I gave him a quick answer in the live chat. Ian or Fiona, if you guys want to expound upon that.
I didn't see the live chat, so please roll the tape.
That's all right. No, I just wanted to say that the reason isn't necessarily the temperature outside. It's really the pattern of human behavior. When it's cold outside, we tend to spend more time indoors, more time spending close contact with many people. And then if one of those persons that's living in your dormitory gets sick, your close vicinity puts you at increased risk for catching the flu. And that's why it spreads so easily and we have these seasons. And there is a sort of protective effect that I mentioned, that when it gets warm, we spend more time outdoors. The influenza virus doesn't do well outside. It does really well when we're in close confined quarters.
That's right. We see, pardon me, RSC.
Go ahead.
We see similar patterns in other infectious diseases. One that I'm most familiar with is meningococcal meningitis, which is a bacterial infection. The periodicity of epidemics of that relate to winter in non-tropical environments and dust storm season in others. And the association is both, as RSC said, behaviorally related, and also relates to the prevalence of other infections that make you more susceptible. So the pattern of seasonal infection is one that is common to a number of different infectious diseases.
And also the start of university term.
Yeah. So many of these conditions are studied in the context of university dormitories, prison populations, and very interestingly, military populations who tend to be in close association with many other susceptible individuals.
Yeah.
And to add to that, I'd like to mention that a lot of schools have been shutting down because of the flu.
Yep.
Yeah. Because it's
and that is preventative.
It's where people mix. You're trying to stop these large congregations of people. And in fact, if you look back to Ebola in Texas, was it? There was talk about stopping sporting events. Because you imagine trying to trace everybody that was at it.
One of the common and repeated epidemic hotspots for meningitis, just irony, a forethought, is the Hajj pilgrimage made by devout Muslims, where tens, if not hundreds of thousands of people congregate over an extended period from many different places in the world and mix together and mix all of the various pathogens together as well. So this is an extremely well-documented issue in infectious disease.
Yeah. Did you want to talk about the vaccine now? We hear a lot about flu.
I did, but I wanted to go sideways just a little bit because I kind of forgot about that. Yeah, I think we need to go on to immunization and and how immunizations work in general and how this vaccine works. But I also wanted to ask, so one, of course, we're going to get to the flu shot, but other ways to prevent catching the flu, of course, are to avoid congregated areas if you can, to wash your hands as often as you can. But are there other things? Do those masks actually work, for one thing? And you see people wearing masks. I've been reading something about this, and I just wanted to know what you guys thought.
Well, I mean, my position on this comes from level 3 bio containment, where masks of that type are used. An N95 mask properly fitted will provide some degree of infection control, because we're dealing primarily with droplet infection. But for a layperson to consistently wear and fit a mask of a suitable type without compromising it, the probability of that is extremely low. So the general precautions that you've listed are excellent, but I'm not a clinician. I will defer to RSC on this.
RSC.
It's okay.
He's wonderful. Anyway,
we can ask him about that later.
If you do have,
I'm sorry, did you not? I must have been muted. In the hospital, you wouldn't see anyone wear a mask. If I'm seeing someone who has the flu, I'm not going to wear a mask because, like Ian said, it's not really protective unless I wear the N95 mask. But that's saved for other things like tuberculosis and other infectious causes. I don't know if there's a lot of research showing and demonstrating that type of personal protection would be effective in preventing the spread. So I really wouldn't want to comment on it either, just like you said, Ian. But I do want to mention the other thing that's really crucial for people out there is if you do catch the flu, don't go to work, don't go to school, don't expose other people unnecessarily to the illness you've already caught. That's the best thing you can do once you've caught it.
Avoid.
Jade went through everything else.
Yeah, avoid elderly people. Avoid children. I had the flu in the 14-15 episode and quarantined myself away from my elderly parents. Reason for this? I'm in good health and I'm a robust, relatively young person, and it floored me for about 10 days. My parents are in their 80s, and it almost certainly would have killed both of them, as well as my great niece and nephew. So getting proper behavior if you are infected and prevention proactively may not be a huge benefit to you, but it will be a huge benefit to those people who are exceptionally vulnerable to this condition.
Can I also add in something else? And it's very old-fashioned, this. Use a bloody hanky. If you're going to sneeze or cough,
cough into your shoulder.
Yeah, cough into a hanky or sneeze into a hanky.
And something I do when I find a sneeze coming suddenly is honestly, and I've seen people look at me and laugh, I'll just grab my shirt and stick my head down toward my chest and sneeze. I'm just not expressing it out to the world. I mean, I'm not saying it's not going anywhere, but it's just, but I'll do that. And I've had friends of mine look at me and laugh. And I said, well, I didn't have time to grab a Kleenex or anything. I just didn't want to, sneeze in everybody's direction. So yeah, or as RC said, sneeze into your shoulder, do something to try to not project. I think some of you out there have probably seen those videos where they show the droplets that travel when you sneeze, and you don't want to do that to other people. And even if you're sick. And about the elderly people there, those people, you guys know, I'm 67. I'll be 68 in May. So this is of concern to me, even though as far as I know, I've never had the flu.
Yep.
Now, what my doctor said, I was just going to say, what my doctor said to me once, I told him, I've never actually had the flu. He said, you may have had the flu. You just didn't have the standard symptoms, or you didn't have it very severely. Just to say I've never had it doesn't mean I've never had it. It just means I've never had a full-blown flu. I've never had anything that I could distinguish from a cold. But that doesn't mean that I can't get it.
Yeah, here's the thing, though. Quite often you will hear people saying, oh, nah, I wasn't well at the weekend, I had the flu. They didn't have the flu, they had a cold.
Yeah.
Because in most cases, when you have the flu, by God, you've got the flu. I was flattened for a fortnight as a teenager with the flu. It was misery. Everything was horrible to do. Even having to get out of bed to go to the bathroom was hell on earth. It was awful because it hurts so much just to move. When you have flu, you've got flu. And too many people will say they've had flu when they've just had a cold. But also, people will soldier on into work with a milder form of flu, with thinking it's a cold. And often in a normal year, that isn't too much of a problem. But in a pandemic year, and in a year with a strain which is so lethal, then if you get the symptoms of a cold, play safe, take sick time. I
think that's a very important point, because as I said, I've never had what Fiona had. I've never had this full-blown flu. And I'm 68 years old. I've never had the flu to my knowledge, at least not in the sense that Fiona's talking about. If you have it, it floors you. Never happened to me. That doesn't mean that some of my colds in the past weren't mild forms of the flu. And so don't take any chances. If you have a cold, you think it's just a cold, stay away from everyone. But not only that, you don't want to add to anyone's infection. They've got the flu. They don't need your cold on top of it.
Yeah.
I don't, so just please be careful this time. And like I said, if you are like me, and I've run into people like me who've never had the flu, don't think that you can't get it just because you've never had it. That's not the way it works. It's already been mentioned how this thing is a moving target. It constantly changes. It constantly evolves. And I keep thinking one of these days I'm going to get the flu and it's going to totally floor me because it'll be my first flu ever. But as I said, undoubtedly I've been infected with flu germs because relative flu virus, okay? Because how do you avoid it in society? It's just never floored me. That doesn't mean it can't floor me. So don't assume that if you've always sailed through like I have, that you're going to continue to sail through.
Yeah. I kind of also add in, when it comes to the transmission, I'm just going to read, now this is just straight off Wiki, I'm going to read it out. Because the virus can persist outside of the body, it can also be transmitted by contaminated surfaces, such as banknotes, doorknobs, light switches, and other household items. The length of time the virus will persist on a surface varies, with the virus surviving for 1 to 2 days on hard, non-porous surfaces, such as plastic or metal, for about 15 minutes in dry paper tissues, and only 5 minutes on skin. However, if the virus is present in mucus, think aerosol droplets, this can protect it for longer periods, up to 17 days on a banknote.
Yep. One of the reasons for that, and Fiona, it's an excellent point, is as a virus, the flu particle is not metabolically active. It is not alive unless it is in concert with a host cell that is also viable, which means it can act like a seed of infection.
Yeah, and also because it's got a bit of the host membrane on it, that's an extra little bit of protection. Remember when we said about it's got this lipid coating on it? And that's protecting it as well. Yeah. So it can be pretty persistent. If you compare it to there are certain bacteria that can't even last that long.
Yeah, that's right.
The two that come to mind are syphilis and gonorrhea. Syphilis cannot survive outside the human body, and gonorrhea survives for a very short time outside the human body. And here we have flu virus on a banknote for 17 days if it's inside a booger.
Yeah. So wash your hands more often than you think you have to is the key to there.
Yeah, because you pick it up in your hands and then you touch your face.
Can I also point out that there have earlier in the flu season, I think around December, there were some newspapers and magazines that were having little editorials, I guess you'd say, on how to properly wash your hands. And I know this might sound silly to some people. Well, we've been washing our hands for years. What do you mean? Well, there's a difference between just, quickly washing your hands with soap and running them under water and thoroughly washing your hands and making sure your nails are clean. Now, I don't know if RSC as a physician could talk about this or not, but if you are at all suspicious that maybe you're not washing your hands right, look it up. There are videos on YouTube about how to properly wash your hands. And when you have a flu season like this, or any flu season, any cold season, learning how to wash your hands properly is probably a good idea. I learned some things from some of the stuff that I was reading, and I realized that I was being far too quick with washing my hands.
Yeah. I hate to point this out,
if
people.
I could jump in.
Can I just point out here that as Jade was talking about that, I am sitting guiltily picking horse shit out of my fingernails.
So RSC, if you could,
Yeah, if I could just jump on that last part where you guys were talking about things to avoid, like touching your face. Yeah, don't pick your nose either. That's not going to help with the influenza infection. But in all seriousness, we do have to wash our hands appropriately. Things like the influenza virus, just because you rinse water over your hands that's not going to take care or provide any protective effect. You have to use warm water. Cold water is going to be much less effective. And B, you have to use some type of sanitizer or soap. Soap is very good, but you do have to put a little elbow grease in. Make sure you cover all your digits in between your fingers. Up to the wrist is generally what's considered appropriate for hand washing. And in fact, I mean, one of the first courses I got in medical school was a 30-minute lecture on how to wash your hands. That's how important it is.
Yeah. So a surgical team prior to any surgical intervention will typically spend, I don't know what it is now, 15 to 20 minutes scrubbing.
Yep. That's about
and
what
that
it
means,
is.
that means scrubbing with a surfactant.
Every square inch of surface area, basically up to your elbows for surgeons, but you guys at home don't have to do anything like that. You will get a protective effect for influenza and a lot of other illnesses if you do it appropriately up to your wrist. And the one thing I did also forget to mention, your cell phone. Get some alcohol pads and clean that thing. It will contain viruses and bacteria from the toilet, from pretty much anywhere you go, anywhere your hands go. So do yourself a favor, pick up some wipes and clean it every other day. Clean it every day.
And I'd like to add, clean your computer keyboard as well. I have 90% alcohol here, and I put it on a little a cotton pad, not a cotton ball because they're too absorbent, but I put it on a cotton pad and I run it across my keyboard constantly. I mean, not constantly, but every other day or so, like you said. Your keyboard and your mouse pick up all kinds of crap. And don't forget your even things like your steering wheel in your car. I actually got pink eye because I borrowed a friend of mine's car.
You?
What was she doing in the car?
No. She had pink eye, and she apparently rubbed her eyes and then put her hands on her steering wheel. I borrowed her car, put my hands on the steering wheel, and apparently rubbed my eye.
Mm-hmm.
Because we do that. When I used to work at a college cafeteria, and the guy who taught sanitation to the food service students, he had this thing where he would hand out this piece of paper to everybody and have them sign it. What they didn't know is he had it embedded with this particular substance, which was harmless, but would show up under ultraviolet light. Because he was trying to teach these students that you have to be very careful for hygiene if you're going to be cooking for people. And what he'd do then is he'd then tell them what he did, and then he'd ask for volunteers to come up, and he'd shine their black light on them to see where they had touched themselves.
Mm-hmm.
Yeah.
And there was often much hilarity because somebody would have inevitably be scratching their crotch. But it's a really good lesson. I watched it once, and it's a really, really good lesson about how we unconsciously touch ourselves all over.
When I think about you, I touch myself. I touch myself. Sorry, it's a song from years ago. But
doorknobs and other things would fall into that same category of things to clean, things that you're going to touch, TV remotes, etc., etc.
I was going to add to
that same potential.
One of the things he did too is he took Petri dishes and he took samples from every doorknob and every handle in the cafeteria.
Yeah.
And I said those are great lessons. No, I'll never forget that. I mean, I wasn't even in the class, but I'll never forget that.
You won't eat in a cafeteria again. Don't do it. Just don't do it. I was going to ask Ian now to take the floor and talk about the vaccine.
Yes.
Reading today, and from decision of what strain to go for to the mass production of the vaccine, I think takes 6 months. So, I mean, they have to try and predict ahead of time.
Yeah.
And so I wanted him to talk about how they go about it and why there is such a problem. That takes us back to the segmented genome business and RNA and all the rest of it. Ian, would you take the floor and just whiffle on and keep going? Because I'll go to the toilet.
I will do my very best. I'll try to avoid whiffle, and I'll try and get some key points in there. So to recap, the flu virus is an RNA virus, which means it does not copy itself with great fidelity, which means that genetically it changes very quickly. And effectively, there is no pure culture of the seasonal flu. It is a moving target. Throughout, as an ongoing thing, flu outbreaks are monitored by local health authorities and the World Health Organization. As a result of this, the infective nature and pathogenicity of individual flu outbreaks can be assessed. Based on this, a selection of flu subtypes, usually 3 of them, are included in the seasonal vaccine. To manufacture the vaccine takes between 6 months and a year under the best possible circumstances. And we're talking about manufacturing from the genetic level up to a scalable process which can produce tens, if not hundreds of thousands of doses. So there is always a lag between the nature of the identified worst pathogens, usually 3, and the generation of the eventual vaccine. So a process of global surveillance coordinated by the WHO identifies the most probable flu subtypes. And I think the current seasonal flu contains the Phuket outbreak strain, the Bangkok outbreak strain, and 1 other, all assessed, to repeat myself, based on their infectivity and pathogenicity, as well as the overall number of cases. So based on that data, a selective process is undertaken, and the strains that are most probably going to seed the remainder of the world are included in the vaccine. Now, that, I think, addresses Tyler's excellent question about how researchers predict the flu strain that will be in the vaccine. And Drag also asked the question about protective efficacy of the vaccine. The numbers associated in terms of protection, which means avoidance, prevention of the infection for flu vaccine, are typically quite low. This year, particularly 30 to 40%. Normally 60 to 70%. And the reason for that goes back to the physiology of the vaccine and its genetic mobility. The organism changes itself extremely fast, and as a moving target, it's not always hit. What is more significant, though, is that the changes within the neuraminidase and hemagglutinin proteins are limited by the function of those proteins. What that means is, if a change happens that abolishes the function of either one of those proteins, then it's essentially outside of the gene pool for the next generations of flu because it no longer works. So there is a constraint on how variable these things can be. Consequently, vaccination with a vaccine that is not a complete protection, not a complete match to the infectious strain, still has benefits. To unpack that a little bit, a protein like neuraminidase or hemagglutinin presents multiple surfaces, multiple epitopes to the immune system, which can be recognized by multiple different antibodies. While those antibodies may not be 100% effective, they do reduce the impact and infectivity of the virus. So, irrespective of the 30%, 40% protection rates, if you are vaccinated in this season, you may be within that 30 to 40% range, but more probably, if you are exposed to flu, the impact of that will be significantly reduced. And that's why partial protection is valuable. Full protection is not something that is really accessible in the current generation of flu vaccines. And this, to repeat again, goes back to the fact that flu varies at each replication cycle as a function of RNA genomes, which are copied with less fidelity than DNA genome, as a function of the enzyme involved not having proofreading and replacement function. So that goes all the way back to the molecular genetics of the virus, through the infectious process that Fiona has described very nicely, and into the immunology. Now, to be very clear, the antibody response is very effective and is one aspect of the immune system. The cytokine and inflammatory cascades that we discussed earlier in the context of septic shock tend to be mediated more by the non-antibody parts of the immune system, the cell-based responses. So the vaccine itself, to recap, is prepared based on the best possible clinical judgment, coordinated in a global sense. The most likely candidates for either highly infectious or pathogenic flus are included in the vaccine, usually 3 of those. The vaccine allows the body to experience the outside parts of the flu, the hemagglutinin, the H, and the neuraminidase, the N, and prepare an immune response to those prior to exposure to a virus that can reproduce and cause disease and cause the cytokine cascade and inflammatory responses that are typical of the illness. So the vaccine itself, there are a number of different versions of it, but the one people are more aware of is a recombinant vaccine that is grown in embryonated chicken eggs. Why is it grown in chicken eggs? Flu in various variants infects multiple different species, including swine, humans, and avian species. Embryonated chicken eggs are a relatively cheap, relatively accessible source of cells to grow vaccine. But if you infect a chicken egg with a human with a human virus, it does not grow particularly well. But it's a means to cultivate the viruses that are ultimately included in the vaccine. Once the eggs are infected and cultured, then there's an extensive process of purification, which removes the components of the egg, the majority of them, and deactivates the infective potential of the virus grown in those cells. That is usually done as a function of chemical treatment using formaldehyde. Formaldehyde is a preservative and at high concentrations is extremely toxic. One of the things I do as part of my job is to measure in very accurate ways the residual amounts of things like formaldehyde in vaccines. In the instance of the flu vaccine, we are talking about low parts per million of formaldehyde. So as an additive, it is present at vanishingly small amounts, tiny relative to what your body produces naturally and tiny relative to what you consume on a near daily basis.
What can I just ask what numbers they are? Because I seem to remember that the body naturally produces something like 1,500 milligrams a day.
Yeah, something like that, Cliff. It varies depending upon diet and metabolism. But in terms of dosage in an individual vaccine, we're talking about parts per million. So that is 0.0 0.0000001
of a milligram.
Yeah, very small amounts. The primary components of the vaccine are the H and N antigens. Those are administered to the body in a formulation that cannot cause productive flu infection, and that is a very important aspect. People who say, I was given the vaccine and then developed flu, are not accurate. I'm sure they're not lying. They think this has happened. But one way that the vaccine is effective is by eliciting a limited array of symptoms based on the immune function, the immune response to the vaccine. So if it works properly, you'll probably feel chills and you'll feel a little bit achy, but this will last for 12 to 24 hours most often. It does not turn into a productive infectious process that will last for a week to 10 days or longer.
Is this the idea that you get a small dose of it, which triggers your immune system into fighting later?
Yeah, exactly. It's like showing your army a picture of the enemy so that they know how to react when they eventually see that opposing force. The critical aspect is that the antigens are the right antigens are delivered, and that those antigens are delivered in a way that can be recognized and responded to by the immune system. I think one of the critical aspects of this particular vaccine is why it has to be developed multiple times, and the reason for that is the variability of the target. And again, that reads into why it is not as effective, for instance, as the vaccine against meningococcal meningitis, or perhaps a better example, pneumococcal disease. The reason being RNA genome that copies itself very poorly, a moving genetic target.
And the shuffling of the segments as well.
That's right. So as Fiona indicates, the genome exists in multiple different segments, and co-infection with multiple strains or multiple forms of the virus from different species can transition or can exchange these segments to produce a virus that is novel, I dare say, containing new information that is not effectively recognized by the host. And it's in occasions like that where you tend to get a virus that's both highly infectious and, to a greater degree, highly pathogenic. It's a moving target. It takes time to generate the vaccine. The vaccine has historically been grown in embryonated eggs, but I need to stress that there are a number of both experimental and marketed vaccines that do not use eggs. In addition to that, the vaccine contains aluminum hydroxide as an adjuvant, which is an irritant that stimulates the immune response to whatever you are administering with it. And that essentially makes the vaccine work. It wakes up your immune system and says, you've got to respond to this challenge. The other component is one that is very commonly viewed as a terrible, terrible toxin, and that is thimerosal. Thimerosal is an antibacterial preservative, and it's present in some, but not all, flu vaccines. The classical anti-vax position is that thimerosal is mercury, which it's not. It contains a form of mercury, and that it causes autism. Unfortunately, no study, and there have been many, and I repeat, no study has demonstrated in a compelling, scientifically valid way that this association exists.
Can I just ask a question? Because there was one of these hangouts quite some time ago, and we talked about vaccines. And in it, it was I'm remembering something from about 3 years ago now, and I think it was something like, it's not like drinking a thermometer mercury; it's a form of vaporized mercury, and that it effectively goes through the body in a very short amount of time, usually the next stool sample.
Yeah.
This is Jade's bit.
I'd like, yeah. I have a link to some research. If anybody wants it, I can bring it up for you. They tested some babies. First of all, the thimerosal is, there are two forms we're talking about that they're confusing. They're confusing methyl mercury and ethyl mercury. So one is far more dangerous than the other.
Everyone?
What?
And as an example for the laypersons in the audience, a comparison would be the table salt that you eat on probably a daily basis, that is sodium chloride. Both sodium and chlorine are extremely toxic, but combined within the sodium chloride salt, they're actually quite tasty and won't do you a huge amount of harm. Similarly,
right. and another
sorry, Jay, carry on.
I was just going to say, and another good example of the difference between methyl and ethyl formulations. Now, just to give you an idea, methyl means a carbon with hydrogens attached, one carbon. Ethyl refers to two carbons that are attached to each other, and then they have hydrogens or other things attached. For example, in methane is CH3, ethane is C2H5. Methyl alcohol is you take one of the hydrogens off and you add an OH group. So it's CH3OH. And then ethyl alcohol then would be C2H4OH. And that may not sound like a big difference to you, but we drink ethyl alcohol. Methyl alcohol will kill us.
Yeah. So the overall point is the chemical form in which an element is administered, I use the term element cautiously there, has a dramatic effect upon the pharmacokinetics, how it distributes and is cleared from the body, and toxicity. And in response to one of Tommy's perennial points, these are things that we're not guessing about. These are things that are studied at the preclinical level, as well as throughout the clinical life of any vaccine of this type. We're not guessing about toxicity and cost versus benefit. These are things that are data-driven.
And I wanted to go back to that paper, which I will look up, Cliff, as soon as I'm done talking. My system acting well now, but it can be flaky, so I don't want to do two things at once. They did a study where they administered a vaccine to babies that had thimerosal in it. I have terrible time pronouncing that. Certain words just kind of do me in. So they gave this to the babies. And you know what? They were surprised by the results, because when they tested the baby's urine, they discovered that stuff just went right through them.
Yep.
They didn't retain it at all. And I will find that link. So this idea, first of all, you said there's screaming mercury. We're not talking about elemental mercury. We're talking about a mercury compound. And even then, when they do talk about the mercury compounds, they get the ethyl mercury confused with methyl mercury.
That's right.
Furthermore, they ignore the studies that show that the body does not retain. The body retains methyl mercury. The body does not retain ethyl mercury.
There are also clinical guidelines that are distributed to physicians describing conditions in which a vaccine with thimerosal should not be given, and those include kidney insufficiency, damage, loss, or disease. There are versions of the flu vaccine that do not contain thimerosal, and those are administered typically to neonates, so very, very tiny people, and to pregnant women.
Oh, and I did want to mention, I do believe that if you have an allergy to eggs, you should not have the regular flu vaccine. I'm not sure about that. Is that true?
No, that's absolutely true. And the reason for that is when you grow a virus in a complex cell like a chicken embryo fibroblast, as many of them are, but not all, there is an inevitable carryover of some of the typically proteinaceous components of that cell into the final product. Again, we're not guessing about the amount or nature of these proteins. These are things that I've personally developed methods to measure and assess. And there are very strict specifications on the vaccine in terms of what is called host cell protein contamination. So this is something that is well understood and is well monitored. However, if you are sensitive to, for instance, egg albumin, one of the proteins in egg white, there is a possibility that some, and again, we're talking about parts per million, some egg albumin protein may be present in the vaccine. And that is why it is unwise to get one if you are allergic to that protein. I should stress, though, that the Novartis vaccine is not grown in eggs. It's grown in avian cell culture, so very similar cellular background, but in a different construct.
Right. Is there anything else that anybody wants to cover, or have we just about covered everything that needs to be covered here? Or any questions from our faithful audience?
I have a question of my own, and I wasn't here, so I've only just picked this up since I joined.
Ask away, Cliff.
There are times when flu vaccines aren't very effective, and the way I understand it is they anticipate it's going to change into this format, and it actually changes in a slightly different one. So some people are okay with it, and other people it's not effective at all. Would that be a reasonable assessment?
Yeah, it's reasonable, Cliff, and it's because we're making a product against a moving target, and this is the challenge. However, even a vaccine against a non-completely matched strain will provide some degree of protection. It will enable your body to make a more prompt and arguably more effective immune response faster when exposed to the wild-type vaccine, wild-type virus. So irrespective of the low overall protective rates, I personally get the vaccine every year.
And it brings up another question, and it's something connected more with me. So I've got agoraphobia. I don't go out very much at all. It's roughly about once in 10 days. I hardly get the flu, largely because I'm not hanging around with people who are infected. Is my immune system going to be a little more deficient as a result of me not mixing with people?
Well, to speak at the basic immunological level, if we're all the same in terms of our immune response, exposure to antigens in the environment will prime your immune system. However, the potential for an effective immune response isn't diminished by absent previous responses. In large part, you're going to be fine, Cliff. Don't worry.
Oh, I'm not thinking that I'm like in danger of anything. I just mean that because I'm not mixing with other people, is my body less likely? Because it's less. I don't know. Let me try again.
Your immune system may be less experienced than people who are exposed to many different things.
For example, one of the things that I've heard, and I don't know whether it's actually true, but it sounds true. I'm very much a layman in such things. I'm considered the person who doesn't know very much at all and is able to ask questions to make So average people who are watching can also, things can tend to go a bit technical sometimes, and that may go over the heads of some people.
Sure.
Anyway, so one of the things that I've heard said is that because we are too clean today, as in, you know, parents don't let the kids play in the mud and stuff like that, and, you know, wipes over everything and Dettol on every single surface and things like that, that effectively many children today, they're not, their bodies aren't given enough stress on their immune system, which means that they're more likely to acquire illnesses. Would that be a fair assessment?
The hygiene hypothesis, yeah, there have been studies into it, and it may have an effect. Yes, from a precise clinical perspective, I'm not really certain about that. One of the reasons I mention it is, irrespective of how clean your house is, if you're dealing with a 5-year-old, they're going to expose themselves to a lot of stuff anyway.
Yeah. Also, I think, I mean, really, that's more to do with the topic of the rise in autoimmune diseases.
Asthma.
Yeah.
It's been tied to asthma.
Yeah, and one of the things that they're saying is basically the immune system isn't getting challenges, so it's bored. And so it goes after other things instead. I don't think anybody's really nailed it down.
Yeah.
It's an interesting point, but in terms of balancing the potential impact of infection versus the possible advantage of exposure to environmental antigens, I would tend to go for wash your hands more often than you think, keep your house clean, and you'll probably be fine. There's one point I wanted to bring up, which is somewhat philosophical and certainly related to public health as well. And that is some recent publications by various church persons in the US saying, vaccinate yourself with the word of God.
Oh, I saw that.
What are the thoughts around the table on that kind of advice?
Bunkum.
It's terrible, dangerous, and absolutely ridiculous.
Yeah.
Might as well let Jesus take the wheel when you're driving down the freeway.
Yeah, and basically, I suppose if you send her some money, you'll get better protection. I'm sorry, you see, that sort of thing really does make my blood boil. We get this sort of nonsense. Yeah. I am not saying don't pray when someone's sick. I mean, it would be unusual for a Christian to say that sort of thing. Oh, don't pray when someone's sick. I'm not saying that. But what I'm saying is this. You use whatever is at hand to deal with the problem that you have. If you have a disease in the community and an effective vaccine, use the vaccine. And this goes on for a little bit, but I'm going to use it again, and people have heard me say this before. There was a wee man on a boat, right? Well, he's on his wee pleasure cruiser type thing, a wee yacht. And it starts to leak. And his pump can't keep up, and he's going down. And some fellow pleasure cruisers come alongside and say, do you need some help? And he says, no, I'm a good Christian. God will help me. And it doesn't matter what they say, he won't accept their help. So they give up and they go away. But they inform the Coast Guard. And the Coast Guard comes out in their wee boat. And they say, come on, we'll throw you a line and we'll tow you to shore. He says, no. I'm a good Christian. God will protect me. They can't persuade him. So they go away. And then a helicopter comes along with a big megaphone-y thing. And they send the wee man down, the wee winch man down. And he says, come on, I'll need to take you off this boat. You're up to your neck in water here. We need to get you off this. He says, no, I'm a good Christian. And nothing they say will persuade him. So they have to give up, and they watch as the boat sinks and the man drowns, and off he goes up to heaven. And he's thinking, what? I'm dead? And he meets God, and he says to God, I've been a good Christian all my life. I've worshipped you. I've gone to church. I've tried to keep your commandments. And when I needed you, you didn't send me any help. And I drowned and I'm now dead. And God said, look, I sent you a pleasure cruiser. I sent the lifeguard launch and I sent you a helicopter. What more do you want? And I think that encapsulates it.
I had a conversation with someone that I will not name here, but he's a frequent outside chat person in G-Man and Ronnie's hangouts. And I said to him, early on in my life, I prayed and was given the message that my job was to use the gifts that God has given me to heal the sick however I can and to help people less fortunate. So that's kind of what I do. And he said, that wasn't Jesus, that was a demon.
What? Well, you see, and we go back to the nautical theme of any port in a storm, don't we?
Oh, God.
So I raised the point with him, and it relates to this conversation that I helped to develop a vaccine called Gardasil, which prevents, very nicely, prevents human papillomavirus. And as a function of that, prevents a large proportion of the cervical cancers that result from that viral infection. But apparently, that is something that is sourced from the dark side rather than the good side. So I struggle with the reasoning involved here. Is it acceptable based on a demonic intervention to prevent disease and death in a group of people? It makes no sense to me.
No, it doesn't. But this is part. I mean, we're sort of wandering off topic here, but this is part of the science denial that is required. And when you speak to people who come out with stuff like that, you can bet your bottom dollar that they will reject evolution. They will be special creation types, usually young Earth creation types. It all goes back to that. And it's spilled over into medicine.
I raised a point, and I try to approach these things without becoming locked into cognitive negativity. The counterpoint was made that throughout human history, natural and self-healing have been highly effective. Why do we need advanced medicine now? And I counter that by saying, well, let's look at infant mortality and life expectancy. And if self-healing throughout human history was so good, why is infant mortality lower now and life expectancy higher now than it has ever been, consistent with access to effective medical care? I've never received an answer to that.
One of the things that always amazes me with... Sorry, go ahead, Jake. Go on.
I was going to say, there are two things I'd like to say about this. First of all, natural remedies are just fine. Things like digitalis from Foxglove. Oh, and you remember that
too much of that will kill you stone dead.
I know. And what about that stuff, that mold that's on bread, that stuff? But the other thing is aspirin.
Yeah.
People were using willow bark and similar things for centuries. The problem is it's actually very dangerous. One blunt way of putting it is it'll rip your stomach out. It can be very dangerous. So along came Bayer, and that's the name of the guy. Yeah, that's where Bayer aspirin comes from. Who removed who removed an OH group from the molecule and replaced it with a CH3 group, which made it less dangerous. So yes, we do have the natural formulation in plants. Go ahead.
Another critical point there is dose.
Yes.
Dose and consistency of dose. Now, a big part... Sorry, Jade. Carry on.
I was just going to say, so you can't get an exact dose with willow bark tea, and willow bark tea can be very dangerous for you. So along comes science, and we've improved on nature. Yay! But I just want to say one other thing about what you were saying before. Is it demonic or not? Well, my response would be that of General William Booth. I don't know if you know who he was, but he's the guy who founded the Salvation Army, and he's a pretty fierce guy. Well, a long time ago, he was criticized because he took money from basically a very wealthy crook. Okay, and he took money from this guy, and he was criticizing the papers. How dare you take money from that man? And General Booth's response was, "I'd take money from the devil himself to do the work for the poor."
Yeah.
So that would be my response to this.
A brief comment about dose and consistency related to medical regulation. We hear very often the evils of the FDA and how they limit and restrict things. Well, one of their primary functions is to make sure that each and every dose of any given medicine, be it a chemical medicine or a biological one, is what it should be at the dose it's prescribed at, and that it is active. None of those constraints exist for alternative medications. So when you take a traditional herbal remedy, you may be getting 10% of the dose or 500% of the dose. And you better hope that the toxicology has been worked out, because there is very little, if any, guarantee that you're getting what you think you're taking.
And let me give you I just remembered this great example. Going back to like the 1970s, I think it was, when I used to read Mother Earth News a lot, and I subscribed to a number of publications like that. I found it interesting, the whole back to the land thing. Well, comfrey, which is a leafy green plant, was incredibly popular. And you'd read in, what is it? Oh God, the Rodale publications. Can't even think of the name of they are now.
Yep.
But and you'd read about how wonderful comfrey was, how good it was for the system, how it had all these great vitamins and minerals and stuff. And a lot of the people, who are going back to the land, they say, well, and comfrey, I grew comfrey. I know it grows great. It grows really fast. It's easy to grow. Great, big, huge, lovely, tasty leaves that you can cook and feed your animals. So there was this big deal. Oh, everybody eat comfrey, eat comfrey, eat comfrey. COMFREY, I think. You don't hear much about comfrey now. One of the reasons is that people were feeding comfrey to their animals, including rabbits, because rabbits are a great source of meat, really easy to grow. But then all these homesteaders that were feeding their rabbits comfrey started to notice that the rabbits had tumors.
Mm-hmm.
Uh-oh. Turns out comfrey, not such a great idea. In small amounts, no problem. But yeah, and people were totally comfrey in salads, comfrey as a vegetable, comfrey tea. Yeah, no, not such a great idea. So yeah, natural isn't. What?
Didn't you have to boil it for 3 hours, drain the water out, and boil it again in order to remove the toxins and actually make it safe to eat?
I don't remember. I'd have to. You know what? Now that we're talking about this, I'm going to have to look this up again, because I'd kind of forgotten about comfrey, because I grew it for a while. I didn't eat a lot of it, fortunately, but so this is the idea that because something's natural, it's good for you, and because it has all these marvelous vitamins and things in it. The thing is, before comfrey became like really, really super popular, people were eating modest amounts of comfrey. But then it hit the Rodale publications, Mother Earth News, and all these other publications, Backyard Chickens, and the rabbit thing that I used to subscribe to. People started going comfrey crazy. You know how people go through stuff like, for a while everybody was eating kale. So comfrey tea, comfrey is a vegetable, comfrey in salads, comfrey everything. So eating way too much comfrey, not a good idea.
Yeah. So dose and being confident that what you are putting into your body is what you think you're putting in, and that has been tested in terms of pharmacokinetics, efficacy, and toxicology is extremely important. And without a regulated medical manufacturing industry, those things are not present.
Listen, guys, I would like to sort of wrap this up, because I am really tired tonight.
Yeah, no problem.
Now, do we want to do another hangout, a sciencey, healthy hangout next week?
Yeah, great. Let's discuss a topic offline. But this has been a great deal of fun.
It has,
Yeah, I
hasn't
think so
it?
too.
Can I just add a question, or a comment onto what was said a few minutes ago, just before Jade went? And it kind of leads into what you were saying, actually, Fiona. I find it interesting how many Christians find the idea of God utilizing people to study, understand, and harness the very nature that God has given us from their perspective is somehow wrong. You know, it's a bit like Fiona said about the sinking boat guy, that practically anything short of Jesus popping up in front of them, laying hands on them, healing them, isn't true, despite the the science and the doctors with their evidence.
Yeah.
During the Black Plague, where medicine was in its infancy in any practical sense, there was a plague, a secondary sociological plague of repentant flagellants.
Yes.
People who, in response to this terrible cataclysm, opted for beating themselves, flailing themselves, and carrying crosses around, and generally debasing themselves physically and otherwise, as an alternative to the very limited medicine that was present there. I see the same reality, medicine, and science denial present today as being an example of exactly the same phenomenon.
And that got to the point, the flagellants got to the point where they were they would invade churches and they would tear down the statues and kill priests. It became very extreme, and as a matter of fact was a subplot in The Name of the Rose.
That's right. As an aside, historically as well, we spoke about the socio-cultural effect of the Black Death plague. There was an ecclesiastical impact as well, such that the rules were changed in terms of who could provide last rites and absolution, quite simply because the Catholic Church at that time was running out of sufficient numbers of priests to do the job.
Right. Because the priests were administering to the ill and catching the disease themselves.
Yeah.
Yeah. Right. Anyway, I think that was actually quite an interesting little hangout to have. We can talk off here what we could do next week. Anybody who's interested, keep an eye open for any notifications until we get ourselves sorted out and in a regular time slot without me falling asleep beforehand and then waking up too late. I want to thank everybody that stuck with us listening to this. I hope we've answered any questions that you might have. If you have further questions, contact any one of us on G+ to ask the questions. And with that, I will say good night, everybody.
Good night, everybody.
Good night.
Thank you for the hangout,
Fiona. Get your flu vaccine.
Yes, and wash your hands. And don't pick your nose.